Yeah, okay. So let's continue. What I would like to at least get through the parameterization section. And as I said, typically you will take a problem and break it down into some smaller components. And there are rules of like how to put things, back together. And one thing, just so the topology files will make sense to you, this may be like the numbering, say, that you'll see in Spartan or on the screen, like a chemist would number. This is the third nitrogen by some IUPAC rules. But really, all those programs care about at the end or the atom types that appear in the topology files and the force fields. So that's also one of the exercises, sort of nice with Mo. It has a little button you just push and labels, and they pop up according to what force field you have. Otherwise, you just use NEdit and open up the file and take a look, which can be a horrible pain as these files get bigger and bigger. So it's sort of a nice feature that this can be done for you. So this N3 is called an NH1, and then there's an NR1, which is this N4. And in this exercise that McCrell put onto the web for you, you'll have to keep your eye on this guy and I think on C6, because those were, by that analogy step, guessed rather poorly, but if you go through the more complete maximum optimization, you actually improve those values and getting them to agree more with the quantum chemistry results. All right, so that's one of those pieces. This is the smaller compound that they're looking at. And this is sort of a typical cutout of part of the topology file. And one thing I want to point out to you, if you haven't already noticed before, they're oftentimes arranged in groups of the atoms. The grouping sort of makes sense. And normally they try to organize things so that within a group, the overall charge, say, is 0 or something that makes sense, like this is where the -1 is coming from or something like that. So in sort of setting up and describing this particular system, for this NR1 we talked about, they decided to go over here and use the assignment that was from a histidine and of an unprotonated ring nitrogen, called it new atom, and it's going to turn out it was a very bad choice, that this will change dramatically as we go through the optimization procedure. The other thing they did was, I think it was this choice of this carbon will also change here, was not also a particularly good one, but maybe a typical one that you could make just trying to find something that's similar that's out there. One of the things that you can do, particularly if you look like you're not quite sure what's going on with, say, this section of the protein, just redefine that as the group, because that gives you a little bit more flexibility to change things, and also it keeps you focused on where the problems were developing as you're trying to assign your parameters. All right, so right now they just assigned everything from something that was in CHARM already. And, oh, I should go back. They did a very good thing. They annotated things. Very important, as I said, that you can trace back what you did and why. Here's the problem that you're going to be looking at this afternoon. When this glutamate attaches to the protein, it attaches through the cysteine residue. So normally there would be another H out here. So just to get things going, we decided to follow sort of their simple rules that they have from going from a methyl group to a methylene group. We took the charge that they had for the hydrogen here. I think it was 0.16. We added it to the charge that the sulfur had in the CHARM cysteine residue. That brought it to 0.7, and we used that, and then kept that whole group still sort of charged correctly, or 0, right? And again, we did what we're supposed to. We annotated it somewhere in here. There should be an exclamation mark saying we did something. Maybe you can't see it. There's an exclamation mark here that says, oh, this is where we took it from. And I put it on also over here. Now, that was just sort of being, following the general rules. Now, depending on your chemical intuition, you may have looked there and said, you know, sulfur, that's a lot like oxygen, right? And some things. So this looks like an ester. So when I've added this whole group to this cysteine, I'm actually, maybe I should be looking at a different fragment. Maybe I should go and take things from the database that have to do more with esters. And it turns out that probably would have been a smarter thing to do, but then we wouldn't have a nice exercise for you guys to go through. All right, so if you now take a look at this, what you will be doing your exercise on is this smaller fragment of this bigger thing. And we do that in part to save ourselves some time so that you can get through the exercises. But you're certainly welcome to take a bigger component and just make this a methyl group here. And since you all know how to run games now, everything that you've done with Spartan, you can run on games and send it to NCSA. It'll run. Please do not do any higher level calculations with Spartan on the local machines. You'll be sitting there to the end of the course waiting for an answer. All right? I mean, it's not that bad, but it's those machines are just not designed really to handle very high level calculations, you know, within an hour or two. Okay, so now it gets to this discussion of the partial charges that are there in CHARM and that you'll accept when you do like a quantum chemistry calculation or you'll compare them. And this is, as I say, because of this closeness between AMBER and CHARM, AMBER does use this fitting, the RSP, the rest fit. The CHARM people claim it tends to overcharge the atoms, maybe gives you bigger dipole moments than you really want. So it is better, they feel, to start with the Mulliken charges and then solvate the group and then do the quantum chemistry calculation on the hydrated things and then take the charges that are coming out of there. I'm going to ask you to decide that for yourself because you'll look at both the ESP charges as well as the Mulliken charges and see which one at the end of the day come out being closer to what actually appears in the AMBER force field. And then this is again some of the fine print that I was talking to you about. So you said you were saying having trouble getting things to be exactly come out the same. Oftentimes the Leonard-Jones interactions you get from quantum chemistry calculation or the interaction energies will be scaled. Sometimes they will also even scale the force constants and the vibrations because in attempting to fit the gas phase ab initio calculations to this whole slew of experimental data, they just notice maybe it's easier to do this. But sometimes this number may change, so you have to really ask them whether it was used in 22 versus in 27. All right. Here's just a note of caution that McCrell put in, so I put it here too, is that you know, you shouldn't just go around changing at will the level of your quantum chemistry that you do. I'm having you do semi-empirical for the tutorial in class because it is fast. We don't expect it to be good. We tried to present you or give you an idea of what a more extensive one would be in the handout. But in general, if CHARM is using MP2 or this Hartree-Fock basis set, you should be using those two in your simulations and not semi-empirical methods. As you go to the Merck fields, I think, and the equivalent one I think is called GAF, that's handed out by AMBER, they will use other semi-empirical methods. But again, you should just use the same consistent pattern. I think it's AM1 there. All right, so we've already discussed, you know, just going to use what's in the CHARM database, getting this thing started, the Mulliken populations. Now, as I said, to get to the final charges and the interaction energies, like the distances appearing in the Leonard-Jones, the well depth, you're going to take your molecule and then hydrate it sort of around all the polar groups. So you're really doing this calculation on a hydrated system. You've got to check the dipole moments, go look at the distances the minimum distances sort of agree, and perhaps rescale things. And this is data, again, from Macrel for various non-bonded interactions. You compares the ab initio. This is analogy just out of picking something reasonable out of the CHARM database. And this is optimized. So you've taken these, you've now maybe done one more iteration on them, looked at the experimental data, perhaps rescaled them. And you can see, I think, the analogy at times is actually working pretty well. There are in some cases where it is not working so well. So that's why they were redoing some of these Leonard-Jones parameters. Same way with the minimum distances. This just gives you the overall statistics, and here are the dipole moments that you would get out of this for this molecule using these 3 different methods. And they told you here they have actually scaled the ab initio interactions all by 1.16 to get them. So finally, let's look what happens to the parameters, how they've changed sort of from my initial starting guess by analogy. So remember we had this NR1 value of 0.16. It has now become 0. Minus numbers, 0.31. That's a large change. The same way with this group, it's changed. The methyl group, the isolated methyl group out here stayed pretty much the same. That seems reasonable. But this whole end of the molecule has been changed. And if you look at this end group here, this methyl group, whereas before it was neutral, it now has a slightly positive charge, accumulation of a positive charge out there. All right, this just really summarizes again what all the angles are that you're going to have to be looking at. So here's the overall molecule that you wanted to get. You know, it's not that, this task is not infinite, right? So, you know, and if you're not doing high throughput, you know, I think you can just, you know, be persistent and you'll be able to finish the job. And again, just to remind you that you have to look at any available crystal structure, vibrational data. The calculations, I don't think at this point we even mentioned them yet, although you did it with hydrated systems, there's still yet to have this thing be run in water or in some condensed phase, perhaps in the protein, to be sure that you're getting values that are consistent, which are measured and seen in the databases, say for proteins, which is the PDB. There's another one for the smaller molecules. It's called the Cambridge Structural Database. That's a very good source of information. The only thing is very hard to find something in there. Although it allows you to put in a formula, a name. I know for the whole problem we wanted to give you on the tutorial, we typed in the accepted name and nothing came out. And then I found an article that said it took 30 of these structures from the Cambridge database and did a statistics on it. So that's what we finally were using to give you the information. So they're in there. We just didn't know how to call them, define them. Right? And in fact, there is the Cambridge database on their particular, on Macrel's example. And again, he's just showing you the results of his full quantum chemistry calculation. This is the experimental data, and this was done by analogy, just taking what's reasonable out of the CHARM force field and doing some optimization on those values. And you know, the good thing is if you look at these values, they all seem quite reasonable. So I think the geometries are coming out, you feel pretty confident about them. So just here, these bond lengths are not too bad. All right, and it's sort of about the range that you can accept. And if you look at enough of these, and he's got tables and tables more, and if you go back to their 98 article where they did the CHARM 22, I think for the proteins, and also look at, it's very important to pull off the supplemental things that are in each of the journals, because they have even more tables of this. You get a sense of what sort of tolerances and convergence criteria they're accepting. All right, so let me just summarize what's going on thus far. As I said, you'll do these Leonard-Jones, the non-bonded interactions. You'll sort of, as a basis of those, there'll be analogy. Then you move on to doing Hartree-Fock on these hydrated systems. And when you're done, at the end of the day, and you'll get your charges and the distances, they have noticed that oftentimes the Leonard-Jones parameters tend to be very transferable. So you don't have to, if you're trying to do something in a faster way, maybe you don't have to change the Leonard-Jones parameters initially. Or if you're doing this thing in self-consistency, maybe you change first the charges and then you do the Leonard-Jones. Now back to your question about those other geometric properties like bond lengths, angles, impropers, dihedrals. So on Friday's exercise that you did with games, another quantum chemistry program, it had a very nice feature in there that it would calculate the normal modes. You'll see them in Spartan as well. I think you use Molden to animate them. In Spartan, it's built in. You can just click on them and it'll animate them, and you can get a feel. Like some of them will have a more dominant contribution to a particular bond stretch or a bond angle than others. But you've got to get the contributions from all of them. And you can do that with these quantum chemistry calculations and project onto particular bonds or to angles. The dihedrals, it's not really possible to do that from these normal mode analysis. So what you typically do is you drive those particular dihedrals and you have to calculate the potential surface. And then by looking at that and then the expression that you have, you can start fitting what the periodicity should be, what the barriers are. And again, you'll do that in your exercise today. And you'll see how well it agrees with the single term. And as you know, as you rotate these things, that's just not dihedrals. You're getting electrostatic, other contributions coming in. So it won't be a perfect fit, and it's something that you have to play with. And as I said, their general rule is always to underestimate the barrier to allow you to sample more configuration space. All right. This is a little bit of a tedious table, but I thought since you guys did that exercise yesterday, I went to the web and downloaded this. From their high-level calculations, these MP calculations, MP2 that they had done, they looked at the vibrational and tried to see from the various modes how much of them are contributing to a particular bond. So is there no contribution from this normal mode to that particular bond stretch, or is like 90% of it coming from there? And if you didn't do this, maybe we can ask Marcus if there's a way that we can print this out also with games, because I think it's very instructive for you to see how much any one normal mode is contributing to that bond and to the force constant. All right. As I said, they also sometimes will also scale the ab initio results, and that will be affecting then your bond force constants and your vibrations. This table, again, it was from, I forgot to put it down here. This is again from Macrell's web page. Gives you a feel for that. And let's go back to those one or two that we were talking about, this NR1, NH1 bond. It got scaled from 400 as being the bond force constant, this being the bond length, 1.38, it was changed to 550 and 0.136. Same way here, the quantum chemistry calculation gave this, it was scaled down to this, same way here. But if you do that, then you get a fairly good agreement with the frequencies that are coming out. So by analogy, you would get this, but if you did the optimization, now you're a little bit closer. Okay? Yeah, and this section just discusses the dihedral optimization. Again, as I said, you're going to have to generate the surface, the potential surface. This shows you all the dihedrals. This has several that you have to worry about. So this is more complicated than what I gave, which you guys will look at. And there's actually several different ways to do this. You can make it really a rigid body around one, but you don't want to try and drive all of them at any one time, so you just do one after the other. And then it's a question of what do you do with the remaining fragments to make it easier for the interpretation. And again, this just stresses that these are sort of the typical two quantum chemistry methods that can be used. And you can use either one of these, but then send it off to NCSA. All right, this goes just through what I said, that they'll have to do a full geometry optimization, and then they'll constrain, say, maybe some of the other dihedrals to be at their typical values and drive the other ones, and then rotate them. And voila, you get a surface, say, for that particular one. Now, what's interesting, and he puts it down here, and we said it before, this dihedral that you're seeing, it's the sum of all the, affecting that rotation is the sum of all the contributions. And what was really nice about his study, and this is why I pulled this picture off, he went through then and broke down these curves. And if you look, one is the quantum chemistry, one is the value you'd get through analogy, and one is if you'd optimized it. Now, for me, the good thing is you look at this, and this doesn't look too bad. Either one would have been, I think, reasonable to have worked with here. But in the next slide, he broke down, say, this one curve into its various contributions. So here's the dihedral, is the blue. Here's the electrostatics. Van der Waals is not contributing very much to this, right? And then that's everything else, is the yellow. So it's, you know, it's sort of to get a feel for this. If you haven't done this before, it's a good thing to do at least once or twice, so you can break this down, right? And you can do that out of the, say, charm. You can just ask it to, as you, if you also to drive a dihedral by doing the analog case or the optimized case, then just have it print out what those other values are. All right. I think this is moving on to the next dihedral. There it looks not quite so great. Here's the quantum chemistry. The optimized finally got there, but if you started off with analogy, you would have been off a lot with this one. And as I said, to do this optimized, this normally means you should also, as I said, when you're done, not just use the results based on the small fragments. You need then to do the full simulation and condensed phase and do the population study analysis to be sure what you're seeing is true. Compound 3 looks a little better, or the third, excuse me, rotation. And then here are some of the rules to how to put these things back together to get these things. So most of the time it involves, like, if you take off a hydrogen, what do you do with that charge? You add it to the carbon sitting next to it and sort of build it up systematically. And most of the time it's fairly straightforward. Once in a while there are, you know, several directions that you can go. And if you have chemical intuition, I think it helps you. If your chemical intuition is not that strong, well, then you should check it as you do now with the final full system, that condensed phase. Look at the population studies to be sure that it makes sense. Now, just to give you a feel for what are the effect of choosing either this quantum chemistry method or this one, I also pulled down his data that he did on several of these compounds where he did calculations using both methods and compared them, just to give you a feel for this. Again, this is a rotation that he did with on this bond, and I think it looks very comparable. The Hartree-Fock. Again, the one by analogy was off, but these two now are agreeing quite well. I'll just go through these quickly. You see analogy here was sort not such a good starting point, doing the optimization, and it makes it agree much more with the quantum chemistry calculation. We'll skip that. That's not so interesting. And now we get to the problem that you're faced with in the tutorial, which is essentially centers around the chemistry of the thioesters. Now, as I said, if we had even just taken a pause and said, okay, look, this is more like an oxygen, so these are like esters, we would have thought two things. And depending how much organic chemistry you've had, you may know that these esters have resonance structures. There's also one, it's just like in the peptide bonds in proteins, you can have a resonance structure where this double bond has moved here, and this has a partial negative charge and the partial positive charge here. There are indications in this article which summarizes all the 30-some structures appearing in the Cambridge database, and not a one of them that we could find. It said that along with some other experimental work, and actually has everything you want to know about these thioesters, said that it is actually doesn't have a very strong double bond character. Now, the reason I bring this up is you go on and do your dihedral studies, you can also, if you want, change it and look at this dihedral study, and you may expect that that barrier should at least be higher than the one next to it. But it should it be as high as the one that you have, like to move something that is a stronger double bond nature, or say in the peptide plane? And it's much less. But you can examine that, and this article also contains values for that. I've written out a few of the key things that you'll be looking at. You're going to be looking at the dihedral bond rotation about this bond. So it's this carbon, sulfur, carbon, and then I think this is a CH3 group, one of the hydrogens. And you would expect that should be a very low barrier, and indeed it is. And it'll be less than a kcal, much like it is for most methyl groups. And one of the things that you'll have to check on is this carbon bond and all the angles that are involved here. And would be interesting just to use and edit, open it up, and take a look at ketones in the something with a ketone in the CHARM27 database, and you'll see that they're actually, the quantum chemistry calculations show that indeed it is closer to those. So if you didn't have intuition to begin with, you'll get it at the end, right? But it can save you maybe 1 or 2 rounds of iteration if you spend a little time thinking about the molecule and its fragments before you start doing your analysis. Now, I point out, besides its obvious importance in this interesting metabolic pathway for this essential amino acid histidine, these thioesters, are becoming more and more prominent in the organic chemistry books because they're the role they play in these acetyl coenzyme A cofactors. It turns out these are major players in driving organic synthesis inside of the cell, and so people are spending more time looking at them, and that's why we're also able to the structure, not only structures here, but there's a lot of information about them as you go into the literature and their reactivity. In fact, one of the places that you can find it, if you want more experimental data that's not in the Cambridge database of small molecules, you can just go to the PDB database. And it turns out that in a very popular protein, the photoactive yellow protein, which is used by a lot of people doing these single molecules experiments, it is a thioester that connects the cysteine to the chromophore that's in those photoactive yellow proteins. And again, you can just look into those, and using that as your condensed phase reference point, and look at the angles and things that you would see, and we give that to you also as part of the tutorial. You will also be, I think, either given this or we discuss it, and this I just showed in the previous transparency. Again, I just want to point out that our initial guess, while sort of the logical thing to do and sort of one of the things that one of the techniques they suggest and when you're building up things. If we had thought about it and looked at this more as a ketone, I think we would have redefined what this was as a group and done a better job, or at least defined the group out to here, and would not have to now go through another round of iteration, just like you guys would have to if you wanted to get the best values, right? So let me show you now just the results that we get that are what's in that charm file. These are the atom types. These are the ESP charges and the Mulliken charges. This was a 631 G star star. We decided to do it a little bit more. 1 star would have been enough. And I do point out, though, these are without any waters put in it, because I just wanted to see that, do we really need those waters? I mean, if I can really get away with transferring the Leonard-Jones parameters, if I don't put in the waters, but use the ESP charges instead of Mulliken charges, just how close do I get? And as I said, initially we were pretty bad here because we used that simple rule of adding all the charge of the hydrogen that we took off onto the sulfur. So this should have been about 0.23 if we'd left it alone and added the charge to the other group, which would have been closer to the ESP charge, which I found interesting. And if you look, I think in general there seems to be a slightly better agreement, except for right here at that terminal, I guess this should be actually a CH3 group, in terms of ESP agreement with what's in the final optimized charm values, even though they started with Mulliken charges and you have ESP charges. So I think it's worth playing around, because my motto is to make this as simple as possible, but rhetorically simple and accepted for publication. Right? And I think if you can show this, then you can make a good case for it. This shows you some of the geometric data. These are the ab initio results we had, and this is before and after. You do minimization. Now you put it and solvated it and done a minimization with NAMD and taken a look. And again, I think the values agree quite well. There are a few differences in the angles, but I think it's because we did nothing to the force constants. And had we done, looked at those and adjusted those force constants from the vibrational spectra, we would have also been better in agreement. And these are actually, these agree quite well with the experimental data that's available from the Cambridge database. All right. So this more or less concludes the part that I have on the parameterization through charm. Again, I think we can take some questions now. I don't know about you guys. I'd like to have a break. And then we'll continue with just a very short few comments about these hybrid models. I'll show you our latest results in trying to combine, excuse me, these sort of more knowledge-based approaches of coarse grain with the full atom simulations to fold a protein. And then I'll ask Romy to, Amaro, to show you a movie on the HIS-H system that'll go along with your tutorial. So why don't we first break for questions and then break for food, all right? Okay, just a second. Okay, I can see you, so what's up? Okay, the question is, you know, there are several quantum chemistry packages out there, like Gaussian, GAINS, Spartan, and by the way, each of them has another way of abstracting from the quantum chemistry calculation the vibrational analysis. If you get charm, they comes with it, I believe, a package called MolVib that does this. I think you guys use MolDin, and Spartan just has it as a button you click, right? So should you be getting the same? I think if you do things like the Hartree-Fock, and they're supposed to, that 631G star is supposed to mean something. Those are accepted basis functions that have been published with parameters. You start doing density functional calculations, there you could start seeing some differences, because there may be some differences in like convergence criteria, even in the functional forms. So you do have to check those. Yeah, okay, this is the extent of my survey right now. I just felt like while I was doing this, why don't we take a look? McCrell also mentions rest charges in any of his presentations, and I have a sense that it would be nice if you could do this, because one of the problems is to be able to automate these procedures and to do it for various solvents. So I think this would be great if you could get away with just using the electrostatic charges and then not trying to tune things, starting with Mulliken, tune them to be able to work with the tip 3. That's my own personal comment on this. I would have to ask McCrell what he thinks about that, but this is what the Amber people do. They don't go through this solvation process. They calculate ESP, and the ESP goes into this program that calculates restricted rest charges to give you things that are more symmetric. You know, like normally if you have a methyl group, if you noticed, all the hydrogens on there were the same. So unless there's some reason that you think from interactions or from geometry that there should be a difference, if you have a methyl group somewhere, those charges should all be equivalent. Okay? The guy next to you? Mm-hmm.
Yeah, I'm actually interested in looking at the distribution of environmental uses, which in my case is kind of a phenol in lipid bilayer structure,
Mm-hmm.
just to see how it affects the dynamics of stuff.
Mm-hmm.
I had to start with a quality 5. I made a quality 5, but the problem in my case is I didn't have any structural information on the compound.
Mm-hmm.
All I had was an octanol water partition, which I can see the quality 5 can reduce to umbrella sampling, but then it defeats my purpose because umbrella sampling by itself is such an expensive calculation.
Sure, sure.
So any comment on how good it is that quality 5 information is that? I don't have structure.
So you started with a compound where you had no experimental knowledge about it.
Yeah.
Is that your problem?
Yeah.
And was it so big that you couldn't do a quantum chemistry calculation on it?
So what I did was I used a Hartree-Fock with 60 star level of theory to
Mm-hmm, mm-hmm.
charges. The compound is very simple. It's pentachlorophenol, so it's phenol
Right.
with 5 hydrogen replaced by chlorine.
Right.
But then how do you decide whether
Yeah. Okay, because this is a little bit dry to go through these things. So I think the fact that you can do the quantum chemistry calculation on it is very good, because if you looked at how well the agreement was between the ab initio results and the final optimized values, that's pretty good. Also, optimized means that they've done a calculation in condensed phase, and you got the right population, say, of dihedral angles, and it didn't change, the structure much. So I think you're in pretty good shape. I think if you get good agreement, or if ab initio is all you have, fine, that's a good starting point. My only caveat would be whenever you're dealing with compounds that have heteroatoms in it, like you said fluorine, or like even with sulfurs, you do know that each of them, some of these quantum chemistry calculations, some of them do a better job of handling them than others, because they typically these are bigger atoms. They have the D or maybe even F orbitals, and they present their own challenge from the quantum chemistry side. So the only thing I would caution you to do in addition to that would be to look up other ab initio calculations done on fluorine-containing things. Are people content? Have you used the right state-of-the-art for doing the calculation on your thing? Now, if they tell you you have to go use yet a higher one, and that's not the one that's in CHARM, you know, my feeling is you got to get the right values in there some way. I mean, they're saying stick the same level of quantum chemistry because he's solvating it with water and then doing the next calculation, you know. But you want just the best set of parameters going in there that you can get. It doesn't hurt if you have. Do you have any information on this compound at all, like maybe a heated vaporization or sublimation? Mm-hmm. Nice. Uh-huh. It's not mine. It's just, you know, they do 200 compounds, and this is a comment they make.
So what I started was with the phenol parameters,
Uh-huh.
and I was using Romax parameters.
Mm-hmm.
Romax doesn't have parameters for carbon and chlorine, so I used OPLS
Sure.
for the non-bonded. And it's nice to see that you also think that the van der Waals can be transported from one force field to another. So do you
Right.
think I'm safe in that regime?
Yeah, I mean, it just depends. Is the environment like mostly water? I mean, if you're trying to do this, I only worry about if you're trying to get values from other solvent stuff. But, you know, in general, you know, those class 2 are very useful because they have done it with maybe 200 for the class 1 compounds have been used, but it's 700, 1,000, 1,500. They've been just parameterized to many, many more compounds. It's always worth taking a look. But do remember that if you're pulling off the van der Waals interactions for one system, if they've all been done in methanol, which is maybe a better solvent for your compound, you know, you may have to think, do I have to just rescale some of those for doing the environmental studies? Other questions before we? Okay. This is the procedure that they use for AMBER, for doing, not AMBER, for CHARM, for doing the non-bonded interactions. They are starting with want to use Mulliken charges, but it's Mulliken charges obtained from these solvated systems. And they're doing that. They think they need to have the waters there to get a better idea. Two things. A is to scale it so it agrees with the tip 3 waters. And then the other thing is to give it some environmental effect. Because if you just to do Mulliken charges on a gas phase ab initio calculation, those would certainly not be that good. So you know, I think you really should send and discuss this with Macrel. I mean, you just have to know what they're doing if you want to use their force fields to know why you're getting any differences at all with what they have. Personally, I think it's better to try to get the electrostatic map correct. That will tend then to get the closest approach with other things correctly. So I think it would also maybe help alleviate or maybe make it even easier to transfer things into different solvents. So that's my own personal view, but if I use AMBER or CHARM, I have to sort of understand where they're coming from and try to do something somewhat similar. Okay, that's it. Mm-hmm. what you can do is at the end of the day, you know, I just put in statistical variation. It just depends on how much effort you want to put in it. Sure, you know, like we're done, like with ours, we know the first step was not that great. If you now start looking at, and you say, well, look, it should really compare much better with the ketones that are there, and you start looking at those. You do that, and then maybe you do yet another round. But in this case, there's so much experimental information about the thioester bond. I would much rather use that than try to do a study of everything that's in CHARM. Look, and I also sort of believe that they've done a reasonable, good job. They want to be able to use these for interpretations, to study mechanisms. So I believe that they're consistent. Right, but you can make that part of your consistency study. Like when Macrell was optimizing CHARM 27 for the nucleic acids, one of the things they used was an enormous study that had just been completed of all the various conformations of nucleic acids in some huge database. And they called that their population study. And then when they did their molecular dynamics simulation on a bit longer piece of DNA, they changed the parameters in order to get their population studies. So they changed some of the barriers to agree with what is seen in the database. So they, when the data's there, they do that. Can we have a break? Not just. I gotta get back to my roadmap. Fortunately, my screen is black here, so I don't. It's there somewhere. See that last step? Condensed phase MD simulations, and there's a little arrow going up. Change everything you've done so far, right? Now it's not that bad, but that was the whole motivation for going forth with CHARM 27 for the nucleic acids. Exactly the point that you're mentioning. Fine and dandy to work on these smaller fragments. We realize that's what you can do comfortably ab initio calculations on, but then you want to put them in, you want to calculate a macromolecule. And now all of a sudden all your DNAs are A form and not B form, or there's not this mixture of A and B. Well, then you have done something not quite right. So you have checked the dynamics. Those take, though, very long calculations, though, right? And then you always worry, well, did I sample enough? And then you can get another whole long discussion of, are there better sampling methods? You know, should I be doing histogram, multiple histogram? Should I do multi-canonical? You know, whatever, to be sure that I've sampled well enough with my particular energy function. But that is now a major, I mean, they recognize it, they say it. And that's why, you know, it takes a long time for them to come out with the newest release. So they did that. They saw that was necessary. And so they feel now that they have the dynamics insofar as they can measure equilibrium properties of various conformational states seem to be in agreement with what is observed experimentally. Yeah, that's fine.